skeletal muscles are voluntary, which again just means that they're part of that somatic motor output. You have voluntary control over that. Cardiac muscle, involuntary, and smooth muscle, involuntary. Both skeletal and cardiac are striated, and if you recall from anatomy, what that should tell you is that they have a specific arrangement of myofilaments, particularly actin and myosin, into these things called sarcomeres. Right? So what makes striation are sarcomeres. Since smooth muscle is not striated, it lacks sarcomeres. In all three, the proteins that interact with each other, specifically to trigger muscle contraction, are actin and myosin, in all three of them. And the way they interact in all three of them is identical. So they will interact with each other to generate force, something called a power stroke cycle. What is different in all three of them is how they get the signal to contract. That's different in all three of them. The signals that are generated that trigger skeletal muscle contraction are different than the signaling for cardiac, which is different than the signaling for smooth. So we need to do a couple things to understand this. We have to understand, first of all, how do actin and myosin interact with each other to generate force? That's called the power stroke cycle. It's actually not that difficult at all. We just need to understand some basics about it. And then we want to look at the regulation of that power stroke cycle, and that's going to be different in each of the three. We'll focus on skeletal first. If we still have a little bit of energy left, probably not. We might do smooth. We'll ultimately do smooth muscle, but and cardiac we'll save for when we do the heart, because that's more relevant. Okay. So let's buzz through these slides here. I don't know what, review those right. You're not going to have to do any identifying on the test about the different muscles there, so we've got those there. But what we're going to do is we're going to focus on skeletal muscle. Remember, the organ of skeletal muscle is attached to the bones by tendons. We had these things called origins and insertions. We didn't bother memorizing those. It's really irrelevant for how they work. But when they contract, right, they bring the bones closer together. In fact, the insertion moves closer to the origin. And we talked about flexors and extensors and abductors and adductors and things like that. They just move the body in different planes, right? So different large-scale kind of movements. It's important to remember, and this will become very clear as we study the physiology, muscles only shorten. They don't expand. They can stretch, but they never push on the skeleton. They only pull on the skeleton. So as a result, they have to occur in antagonistic pairs if you're going to do antagonistic action. Right? If you need to do flexion, you need a flexor, the biceps brachii, for example. If you're going to do extension, you need an extensor, the triceps brachii. And they pull on the skeleton in opposite directions. That joint is called, in physics sense, we'd call that a fulcrum. It's a pivot point around which bones move. Some of you going into physical therapy? I think some of you are. I don't know who, but I know some people are. When you take physical therapy, that kind of thing, biomechanics becomes much more important, because there you're looking at the physics of how the body moves in space, right? And that's a larger kind of organ system level kind of thing. Before you can understand that, you have to understand it at the cellular level and see how things work at the cellular level. Now, there is a hierarchical structure to the way muscles are built. If this whole thing is the muscle bundle, if you will, if we looked at it on the inside, we noticed that cells are grouped into these things called fascicles. And we talked about that in anatomy class. And then if you look at a fascicle is a collection of cells. So here we get a cell. And we mentioned that because the communication in here is all electrical, it is important to be able to insulate these cells from each other, right? We don't want all the cells touching each other, because if they did, when one cell membrane got an action potential, if it's automatically touching another one, it's going to get it as well. Right? So in order to regulate the force of contraction and to do what we call recruiting more muscle cells to generate more force, we need to be able to have them insulated from each other. So if you remember, we had endomysium surrounding muscle fibers, perimysium surrounding fascicles, and epimysium surrounding the entire muscle. Again, it's just connective tissue insulation. Some terminology, we often call muscle cells muscle fibers, or myofiber. So if you hear me say skeletal myofiber, that's just a skeletal muscle cell. We often talk about the cell membrane. The technical word for a cell membrane is plasmalemma. So if it's in regard to a neuron, we would say it's the neurolemma. If we're talking about a muscle cell membrane, we'll often say sarcolemma. You may hear me say sarcolemma, you may hear me just say cell membrane. But sarcolemma is just specifically the cell membrane of a muscle cell. Sarcolemmas have potential. You can generate an action potential on a sarcolemma just like you can on a neurolemma. Right? So neurons and muscle cell membranes have the capacity to be what we call excited, to generate action. Okay. Cytoplasm, we may say sarcoplasm if we're talking about that. Now remember, the sarcoplasm, the cytoplasm is just everything inside the cell membrane. Organelles and cytosol. Okay. There are a couple unique, well not unique, I guess unique, modified and or unique organelles inside of muscle cells. First of all, there's some modified smooth endoplasmic reticulum, and rather than functioning in lipid synthesis, which is what most smooth ER functions in, it's just storage, and what it stores is calcium. So there is a lot of calcium outside of cells. We mentioned that there shouldn't be any calcium in the cytoplasm, right? Cytoplasmic calcium levels should be low. And even in muscle cells, they're low in the cytoplasm, but there's an intracellular store of calcium, right? So we take some calcium inside the cell and we store it in the sarcoplasmic reticulum, and that's where it's in high concentration, but out in the cytoplasm, it's still low. Okay. So the way to think about it is it's a ready source of calcium for the cell. Rather than having to rely on calcium from extracellular fluid, we have an intracellular fluid storage compartment, if you will, for calcium, where we can deliver it to the cell real rapidly, okay, because it's got its own storage. The other organelles that are found in muscle cells, I'm not listed on here, but it is an organelle, would be called a myofibril. And a myofibril is just a modified organelle, and it's composed of proteins. So it's a non-membrane bounded organelle.